Sarah, welcome. So you're joining us for the International Conference on Medical Device Standards and Regulation here in Arlington, Virginia at AAMI. And you are a speaker this afternoon in our symposium on new requirements around IVDs, in vitro diagnostics, driven primarily by changes in Europe, right? So before we dive into all that, introduce yourself a bit to folks out there.
So I am Dr. Sarah Chavez. I am working with a writing regulatory company called Criterion Edge. And our role is to provide scientific writing support for companies, both big and small, as they're trying to navigate the requirements for MDR and IVDR. My background, though, I'm a molecular biologist by trade. So I got an undergraduate degree in biochemistry. And then I moved into molecular cell biology. So I've done a lot with genetics and genomics and cancer biology. I've done a lot of IVDs myself, developing a lot of the assays and the protocols for pathology and for actually making diagnoses. So it seemed like a natural transition, having worked for the company and done the regulatory writing side, to actually transition over to serving as the director for IVD Writing Services, just given my background.
Okay, excellent. Well, I know Joe's got a list of questions here. He's going to dive into the details with you, but I want to ask, just sort of set the stage for us. What's driving all of this change? What's happening in Europe and elsewhere?
Yeah, it's a big transition, I think. Previously with IVDD, most companies were pretty comfortable with presenting their own in-house data. You know how well your device performs under laboratory-controlled settings. You have all that data. You've been required to do that all along. The new transition, though, it's giving that real-world data that a lot of the companies haven't necessarily been collecting up until now. Your device may perform exactly as intended under laboratory situations, but what happens when it gets out into the real world? And so that, I think, is the biggest challenge. It's knowing what data is out there and using systematic review of the published data to help you gather that information, but also knowing when it might be necessary for you to conduct your own real-world trial in real patients.
So, Joe, we've been talking about this. This is a real sea change for folks in that space, right?
It is a real sea change for the IVD manufacturers. They're being asked to do a lot of things that they really didn't have to do in the past. Meet new standards and, of course, document their effectiveness or their clinical performance and effectiveness. And the document review is going to be a critical part of that. Yes. So maybe you could tell us a little bit about what is involved in that document review and what are the strategies that an IVD manufacturer might implement to do an effective and sufficient one for regulatory purposes.
So in addition to being a molecular biologist, I'm the daughter of an English teacher. And so over the years, she always told me, document your writing process. You don't go from start to finish in one draft. And that's kind of the same advice I tend to give to our writing clients. It's that you need to be organized with your pre-writing and your pre-work. You need to come into it knowing that your IFU has been finalized, you're clear on your intended purpose of your device, because all of that is going to help you ultimately cast a wide enough net to tell the story. The state-of-the-art section in a typical MDR report is going to be telling you the landscape. So where does your device fit into the broader scheme of things? And that's really what scientific validity in IVD is trying to do. You're trying to set the stage and saying, under what circumstances would I be using this test in a real world setting? Who's going to order it? For what purpose? What are the specific indications for different patient populations? When might it be appropriate and when might not it be appropriate? So that all goes into that pre-work, that pre-writing, coming up with your outline, developing the proper search terms that you're going to incorporate into your systematic literature review so you can cast a wide enough net. What I find is that sometimes the searches, the clients start out very, very broad or very, very narrow. And either way, you're going to either wind up with too much or not enough data. And so working with librarians and people who are trained on actually developing those mesh terms and the sequences of the terminology that we use for our searches is going to be really, really critical. But you have to have those input documents ready to go. So that's what I feel like as a writer, my role is to help translate what the experts, all the companies we work with, they're the experts on their device. We are not. We are the writers, but we're there to help them tell their device's story. So they tell us about their device. We then can translate that into the proper terms to use so that it's not just simply a PubMed search for pregnancy test or COVID test. That's going to come up with literally tens of thousands of hits. You need to know how to refine that down to a manageable number. So really, that's the systematic part of the review. You need to know how to narrow your focus to where you capture the correct data if there is clinical real-world data that's been published. But then you also have to have very set organization and inclusion and exclusion criteria so that you can make sure that it's objective and that it's thorough and it's reproducible. So the other part of that documentation is that methodology, and keeping track of how exactly you did that search is going to be really, really critical. And I find that that's the part that is somewhat mystifying to most people who've never done this process before, and it can be very overwhelming.
So it's not just a broad Google search. It's a structured and methodological search to create the data and information you need. How do you know when you have the right data, the sufficient data, and how do you weed out the stuff that is not going into the file, or at least you're not using it to support your argument?
Yeah, that comes in with a lot of the conversations, the back and forth that we have with the clients. And in many times we encourage them to bring in cross-functional team members. So folks from regulatory, folks from R&D, they're going to know the nuances of the assay. So if you have a particular test and let's say it's approved for an adult population, like in the early stages of a disease, you can use your search terminology to perhaps exclude any articles that are going to come up with late stage disease or pediatric populations. You can design the search in such a way to be very targeted, but at the same time, you then need to have a strategy for screening all those articles. And that can get tricky too, because sometimes you're going to wind up with hundreds or thousands of articles, and you need to be able to look at those titles and abstracts and identify quickly using objective terms why you're going to include or exclude an article, and then document that. So if you read through title number 1 and it's off topic, you need to exclude it as being off topic. If it's, if it's in the wrong patient population, or if they're using serum and you're only indicated for tissue samples or biopsy, then you can exclude that. But you have to track why exactly you're using or not using every single article that you retrieve through that search. And that's the part that requires a lot of organization.
So is it similar to a design control file? You are required to track the results of your searches and all inclusions and exclusions as a result of those searches?
Exactly. And that's one of the biggest gaps we're finding with a lot of the clients who attempt this on their own and then find out that they're a little bit outgunned. They don't really know what they're supposed to be doing. Ultimately, it's that they try to run a PubMed search and either they don't get anything. And, sometimes that is the case. If you have a test, like an HIV test, they've been on the market for 30 years. So, you're going to have a lot of data from 30 years ago, but maybe not so much in the past 5 years. So knowing how to tweak your searches and your search strings to be able to fully capture that data, but also knowing how to handle something that's new and cutting edge, where maybe the analyte isn't even established yet. Then that scientific validity and the proving the association of that analyte with the clinical state is going to be really, really critical. So you want to make sure you capture articles that are more basic science, actually showing that this particular analyte does allow you to make that diagnosis. For something like HIV, those are very established and standardized, is sort of the term that IVD uses to describe those well-established or legacy types of devices. But it's more or less the association of the analyte is established and standardized. But you still have to have clinical data. You still have to go out and search for that in the real world.
When you'd ask about sufficiency, so how do you know when you've struck that balance and you have enough but not too much because of all the excess documentation that creates? I mean, how do you
That is the number one question that we get. Everyone's worried about, is it enough? And so the rule of thumb that we use is that we want to ensure that our data sources are thorough, so that we've searched across multiple databases. We don't just do a quick PubMed search. We use detailed search strings and then search across Embase, Medline, multiple different databases simultaneously to fully capture all that data. You start with that 5-year because you're trying to say what's current and state of the art. So usually you start with that 5-year range, but you may need to go back to 10 or 15 or 20 even, depending on what type of device you're dealing with. And then once you get that information, you may still wind up without a whole lot to work with. That's when you may have to go beyond just published data. You can't always rely on the kindness of strangers to publish data on their devices for you. So now you may actually have to start to design a search or design your own experiments and like a controlled trial. And I know everyone's trying their best to avoid that, but in some cases that may be necessary. That I think is going to be the biggest shock for a lot of the IVD companies.
Are you talking about clinical trials or both laboratory demonstrations and clinical trials?
A little bit of both. Okay. And actually what we find is that a lot of clients now are actually reaching out to us before the device has even been fully manufactured. They're still in their R&D stage to do the scientific validity and to start to conduct those searches so that they can know going into their research and development phase that they actually are state of the art. You start out and you think, I'm going to use this particular primer set to detect this particular pathogen, but is the technology itself evolving into a different area? Are there some particular problems with that particular gene target that you need to be aware of going into it? So by doing these searches up front, it actually helps you with that design phase and that analytical performance. You start to recognize whether or not your competitors are capturing different pieces of data. Maybe you're not collecting a certain data point, but they are. In order to compare yourself to them, you want to make sure that you're capturing the same types of data so that it's directly comparable.
That's a really good point you made there. You said you want to do it up front. And so how early do you start your literature search for this purpose? Because it's like with anything, you want to start things as early in the design phase as possible, right?
And that I usually tell people the earlier the better. Even if you're still trying to decide on which particular primer set you want to use or which particular target you're going to use. A lot of these pathways that you're trying to analyze, there are multiple targets. And if you scan the literature, you're going to find different approaches to making the same diagnosis. Knowing what other people do early on is going to help you make sure that you can make that justification for your risk-benefit ratio long-term. Okay. So we always tell people, go ahead and do it up front, but the thing about systematic literature review, by being so well documented, it is super easy to run a gap search later on. So you may be in the R&D phase, you're not ready to submit for another 6 months or a year. You can do these searches, do the screening and the analysis and the data extraction, and then use that information to inform your design, and then 6 months later run a quick gap search to see if anything else has happened in the past 6 months to supplement your search. Okay. And as long as you're being very methodologically sound and you're keeping track of everything and documenting it, it's very easy for this to be a process. And that's our main message to folks. This is not a one and done report. This is a new process you're setting up. So if it's a hot mess from the beginning and it's very disorganized, you're going to be in a lot of trouble when it comes time for an update. You want to make sure you're starting on a strong foundation and that you haven't set yourself up for the next review being even worse.
Okay, you mentioned both risk and real-world data. Mm-hmm. Are there requirements around the documentation in the EU IVDR about ongoing documentation? You mentioned this is not just, as you said, not one and done. This is an ongoing life cycle process that you'll have to continue.
Yes. So you'll have a performance evaluation plan in addition. And the plan is just telling you, how do you plan on continuing to capture this types of data moving forward in the future? You're not going, if you could anticipate every single risk associated with an incorrect cross-reactivity or an interference going into it, you would be able to avoid it. But you can't. And you don't want to design your searches to be so targeted that you would miss those in the the event that they do come up 5 years from now or 10 years from now. So designing your searches in such a way that they can be broad enough to capture new changes or new published information about your device or your class of devices is going to be really helpful.
Okay, so that you mean back to risk again, because we didn't actually mention that at the first. We talked about effectiveness or performance, but the document search is also looking in advance to make sure you're doing risk, because the IVDR also has new and stronger requirements for risk management for IVDRs.
Yeah,
Is
and
that correct?
it is going to be different than what we typically associate with risk in the medical device world. Usually a medical device fails and something directly happens. In this case, it's that sort of indirect risk, and it is going to be higher for the different risk classes. You know, if you have an infectious disease that you're testing for, there's a risk not only to the patient but also to the general public in the event that there's an incorrect test result. Some tests may require more invasive follow-up testing if it tests positive. So being able to justify your false positives and your false negatives, your sensitivity and specificity, all those different features of your test in the context of the broader clinical diagnostic scheme. That's really what you want to focus in on and capture the data that's going to help support the risk-benefit ratio.
All right, Joe, so we've got to get her to the symposium. She's got a whole room full of people waiting on her.
We do. Sarah, thank you very much. You've covered a lot about documentation and what the IVDR is requiring. Along with documentation, there's the risk management issues, the new risk management requirements, the new quality systems requirements, and of course the clinical trials requirements. So there's a lot of information that IVD manufacturers are going to need, they're going to have to develop, and they're also going to have to share with each other. And so thank you very much for helping us here.
Yes, we're happy to be here.
Thank you for being here.
Okay, thank you.